Gastric acid is a digestive fluid, formed in the stomach. It is composed of hydrochloric acid as high as 0.1 m, potassium chloride and sodium chloride. The acid plays a key role in digestion of proteins, by activating digestive enzymes, and making ingested proteins unravel so that digestive enzymes break down the long chains of amino acids. Gastric acid is produced by cells lining the stomach, which are coupled in feedback systems to increase acid production when needed. Other cells in the stomach produce bicarbonate, a base, to buffer the fluid, ensuring that it does not become too acidic. These cells also produce mucus, which forms a viscous physical barrier to prevent gastric acid from damaging the stomach. Cells in the beginning of the small intestine, or duodenum, further produce large amounts of bicarbonate to completely neutralize any gastric acid that passes further down into the digestive tract. Gastric acid is produced by parietal cells in the stomach. Its secretion is a complex and relatively energetically expensive process. Parietal cells contain an extensive secretory network from which the gastric acid is secreted into the lumen of the stomach. These cells are part of epithelial fundic glands in the gastric mucosa. The pH of gastric acid is 1.5 to 3.5 in the human stomach lumen, the acidity being maintained by the proton pump H+/K+H pace. The parietal cell releases bicarbonate into the bloodstream in the process, which causes a temporary rise of pH in the blood, known as alkaline tide. The resulting highly acidic environment in the stomach lumen causes proteins from food to lose their characteristic folded structure. This exposes the protein's peptide bonds. The gastric chief cells of the stomach secrete enzymes for protein breakdown. Hydrochloric acid activates pepsinogen into the enzyme pepsin, which then helps digestion by breaking the bonds linking amino acids, a process known as proteolysis. In addition, many microorganisms have their growth inhibited by such an acidic environment, which is helpful to prevent infection. Secretion. Gastric acid secretion happens in several steps. Chloride and hydrogen ions are secreted separately from the cytoplasm of parietal cells and mixed in the canaliculi. Gastric acid is then secreted into the lumen of the oxyntic gland and gradually reaches the main stomach lumen. The exact manner in which the secreted acid reaches the stomach lumen is controversial, as acid must first cross the relatively pH-neutral gastric mucus layer. Chloride and sodium ions are secreted actively from the cytoplasm of the parietal cell into the lumen of the canaliculus. This creates a negative potential of -40 MV to -70 MV across the parietal cell membrane that causes potassium ions and a small number of sodium ions to diffuse from the cytoplasm into the parietal cell canaliculi. The enzyme carbonic anhydrase catalyzes the reaction between carbon dioxide and water to form carbonic acid. This acid immediately dissociates into hydrogen and bicarbonate ions. The hydrogen ions leave the cell through H+/K+H base antiporter pumps. At the same time, sodium ions are actively reabsorbed. This means that the majority of secreted K+ and Na+ ions return to the cytoplasm. In the canaliculus, secreted hydrogen and chloride ions mix and are secreted into the lumen of the oxyntic gland. The highest concentration that gastric acid reaches in the stomach is 160 mm in the canaliculi. This is about 3 million times that of arterial blood, but almost exactly isotonic with other bodily fluids. The lowest pH of the secreted acid is 0.8, but the acid is diluted in the stomach lumen to a pH between 1 and 3. There is a small continuous basal secretion of gastric acid between meals of usually less than 10 mg/h. There are 3 phases in the secretion of gastric acid which increase the secretion rate in order to digest a meal. The cephalic phase, 30% of the total gastric acid secretions to be produced is stimulated by anticipation of eating and the smell or taste of food. The signaling occurs from higher centers in the brain through the vagus nerve. It activates parietal cells to release acid and NaCl cells to release histamine. The vagus nerve also releases gastrin-releasing peptide onto G cells. Finally, it also inhibits somatostatin release from D cells. The gastric phase, About 50% of the total acid for a meal is secreted in this phase. Acid secretion is stimulated by distension of the stomach and by amino acids present in the food. The intestinal phase, the remaining 10% of acid is secreted when chyme enters the small intestine, and is stimulated by small intestine distension and by amino acids. The duodenal cells release enteroxytocin which acts on parietal cells without affecting gastrin. Regulation of secretion. Gastric acid production is regulated by both the autonomic nervous system and several hormones. The parasympathetic nervous system, via the vagus nerve, and the hormone gastrin stimulate the parietal cell to produce gastric acid, both directly acting on parietal cells and indirectly, through the stimulation of the secretion of the hormone histamine from enterochromaffin-like cells. Vasoactive intestinal peptide, cholecystokinin, and secretin all inhibit production. The production of gastric acid in the stomach is tightly regulated by positive regulators and negative feedback mechanisms. 4 types of cells are involved in this process: parietal cells, G cells, D cells and enterochromaffin-like cells. Besides this, the endings of the vagus nerve and the intramural nervous plexus in the digestive tract influence the secretions significantly. Nerve endings in the stomach secrete 2 stimulatory neurotransmitters, acetylcholine and gastrin-releasing peptide. Their action is both direct on parietal cells and mediated through the secretion of gastrin from G cells and histamine from enterochromaffin-like cells. Gastrin acts on parietal cells directly and indirectly too, by stimulating the release of histamine. The release of histamine is the most important positive regulation mechanism of the secretion of gastric acid in the stomach. Its release is stimulated by gastrin and acetylcholine and inhibited by somatostatin. Neutralization. In the duodenum, gastric acid is neutralized by sodium bicarbonate. This also blocks gastric enzymes that have their optima in the acid range of pH. The secretion of sodium bicarbonate from the pancreas is stimulated by secretin. This polypeptide hormone gets activated and secreted from so-called S cells in the mucosa of the duodenum and jejunum when the pH in duodenum falls below 4.5 to 5.0. The neutralization is described by the equation HCl plus NaHCO3 NaCl plus H2CO3. The carbonic acid rapidly equilibrates with carbon dioxide and water through catalysis by carbonic anhydrase enzymes bound to the gut epithelial lining, leading to a net release of carbon dioxide gas within the lumen associated with neutralization. In the absorptive upper intestine, such as the duodenum, both the dissolved carbon dioxide and carbonic acid will tend to equilibrate with the blood, leading to most of the gas produced on neutralization being exhaled through the lungs. Role in disease. In hypochlorhydria and echlorhydria, there is low or no gastric acid in the stomach, potentially leading to problems as the disinfectant properties of the gastric lumen are decreased. In such conditions, there is greater risk of infections of the digestive tract. In Zlingera-Ellison syndrome and hypocalcemia, there are increased gastrin levels, leading to excess gastric acid production, which can cause gastric ulcers. In diseases featuring excess vomiting, patients develop hypochloremic metabolic alkalosis. Pharmacology. The proton pump enzyme is the target of proton pump inhibitors, used to increase gastric pH in diseases that feature excess acid. H2 antagonists indirectly decrease gastric acid production. Antacids neutralize existing acid. History. The role of gastric acid in digestion was established in the 1820s and 1830s by William Beaumont on Alexis Street Martin, who, as a result of an accident, had a fistula in his stomach, which allowed Beaumont to observe the process of digestion and to extract gastric acid, verifying that acid played a crucial role in digestion. See also, stomach, digestion, gastroesophageal reflux disease, discovery and development of proton pump inhibitors, acidic mouth, notes. External links, the parietal cell, mechanism of acid secretion at vivo.colostate.edu, First Principles of Gastroenterology. Chapter 6. Salnaby J., Hunter H. The Stomach and Duodenum, Gastric Juice at Dawlins Medical Dictionary, Physiology at MCG6S6CH49.